Today's lecture is over microbial genetics. So microbial genetics, of course, is also genetics, at least a subset of the field of genetics. And so as a consequence, of course, there's a lot of overlap between microbial genetics and genetics as you may very well have learned it in your introductory biology class. But there are certain things about microbial genetics that make it stand apart. And what I'll do today is try to consider some general principles of genetics and then also some general principles of microbial genetics. And in that way, I won't be duplicating what otherwise is the lecture for this class. So microbial genetics. So looking through the book, naturally it starts by talking about genetics in a more basic, more than just microbial genetics sense. And they start early on by talking about genotype versus phenotype. And it's very much worth talking about the difference between genotype and phenotype. And for microbiology, especially as studied molecularly in the lab, the differences between genotype and phenotype are very important. But also, as we study microbiology in terms of diagnostics, the difference between genotype and phenotype is important as well. So this is more than just a fundamental, not very applied aspect of microbial genetics, trying to understand this difference between genotype and phenotype. So phenotype, as I like to say, basically describes everything you can see about an organism, using the word see rather generally. So it's all aspects of an organism, except for what is passed on from parent to child in the course of reproduction. So when we think about genotype, usually it's DNA that comes to mind, although in microbiology there are certainly a number of, I'm hesitant to use the word organism, although I do like to use the word organism, but there are a number of different entities that in fact use RNA as the hereditary material rather than DNA. Specifically, these are viruses. The reason why I'm hesitant to call them organisms is because although I don't mind calling them organisms personally, there are a number of people that in fact do like to not refer to viruses as organisms. But nevertheless, viruses have, by definition, genetic material, and some of those viruses, the genetic material consists of RNA rather than DNA. And although there are some differences in the use of RNA as a hereditary material rather than DNA, there's also a lot of similarities, and I'm not going to go into the differences in this lecture. But nevertheless, that DNA, or that RNA in the case of some viruses, represents genotype. And every other aspect of the organism represents phenotype. And so it's important to, therefore, at least in my opinion, to wrap one's mind around what exactly one means by phenotype. And I'll do this from very much a microbiological perspective, since in fact what we're covering here is microbial genetics. So when we talk about phenotype, we've considered all sorts of aspects of the anatomy of a bacterium. And so starting from the outside going in, whether it has a capsule, that's phenotype, or a slime layer, that's phenotype. Whether or not it has flagella, or where those flagella are located, or how many flagella there are, that's phenotype. Whether it has a thick cell wall or a thin cell wall, that's phenotype. Whether it has a second membrane, an outer membrane, as well as an inner membrane, or just a single plasma membrane, that's phenotype. Whether it has a waxy outer envelope component like you see in the mycobacteria, that's phenotype. All of the biochemical processes that we consider in microorganisms, that's phenotype. Everything that we're going to be doing in the laboratory in this course represents an analysis of phenotype. Now that's not to say that we couldn't be also looking at genotype. When we're doing PCR, polymerase chain reaction, that explicitly is looking at genotype. When we do DNA fingerprinting, that explicitly is looking at genotype. When we sequence a gene or a whole genome of an organism, all of its DNA, or RNA in the case of RNA viruses, that too is genotype. And it is certainly possible to use genotype as a means of diagnosis, of a means of figuring out what kind of organism is present. And in fact, using genotype can be more precise in determining what's present than when using phenotype. On the other hand, we have a lot of tradition in microbiology associated with using phenotypic characterizations. And there is an important difference between genotype and phenotype in their powers. So when we're using phenotypic characterizations, this is something that allows us to determine especially the type of organism that we're dealing with. So determining the species of organism that we're dealing with. And certainly that's possible from a genotypic perspective. In fact, sequencing the 16S rRNA genes, which are genes that give rise to RNA, which makes up part of the small subunit of a ribosome. This is also a way in which we can distinguish among different types of organisms at a species level, because the organisms found within a given microbial species, a given species generally, they're going to have 16S rRNA sequences that are more similar to each other than the 16S rRNA sequences that are associated with other types of species. In eukaryotes, I should note, it's not 16S rRNA, but instead 18S rRNA. But they're pretty much, I mean, not this identical, but they're equivalent things. That's the word. So we use phenotype a lot to do diagnoses. We do all sorts of phenotypic analyses. We could be doing genotypic analyses instead. But that just takes essentially different equipment and to a degree different training. On the other hand, it is very useful to do genotypic analyses if we're trying to establish clonality. So when you have an outbreak of a disease, it is important in trying to control that disease to figure out whether or not there is a point source of the disease versus it being a more general aspect of the community within which the disease is found. Because if there's a point source, if an infectious disease is coming from one place, such as one kitchen in one restaurant, then it is much simpler often to control that disease than if there are many sources. And so it is possible to, in some cases, determine whether something is coming from a single source, if indeed a single organism is being introduced, a single type of organism, a single strain of organism is being introduced at that point source. On the other hand, it's possible that the disease is not easily traced back to a point source, but nevertheless, it is the same organism that is affecting different people. And in this case, again, an establishment of clonality, showing that they have the same genotype, which is the easiest way to establish clonality, can be very useful. Because the alternatives is that something more general is going on. If you have multiple infectious agents of similar type entering into a population, and they're not clonally related, it probably means that some kind of procedure is being messed up. So for example, you could be in a hospital and you're having problems preventing the contamination of patients with environmental bacteria. In that case, it might not be identical bacteria that are getting into these patients, but instead simply the act of allowing different bacteria to be exposed, or these patients to be exposed to different bacteria, some of which are because they have the potential to cause infections, some of which are causing infections. So genotype is obviously an important aspect of microbial genetics. It underlies all of phenotype, But phenotype has its place as well in studying microorganisms, because ultimately, this is the way I think about the world, ultimately, if you're going to be considering the ecology of an organism, it's not really the genotype that interacts with an environment, which is how we define ecology, interactions with environments, but instead it's an organism's phenotype that interacts with an environment. And therefore, in order to understand the potential for an organism to exist within an environment, exploit that environment, to persist in that environment, we have to understand its phenotype. We have to understand more than just genotype. And importantly, when we have an infection, the environment of the organism that is doing the infecting is in fact the body that it's contained within. So all of the study of infectious disease ultimately is the study of microbial ecology, although people often don't think about that, mainly because people who are trained to study pathology and medical microbiology typically are not highly trained also in ecology. But nevertheless, if we want to understand how an organism functions, yes, it can be helpful certainly to know their genotype, but ultimately what we want to understand is the phenotype. If we're studying the genotype first, we want to understand how we go from that genotype to a given phenotype, and therefore to a given ecology for the organism that we're interacting with, or if you may, in medical microbiology, the pathology of the organism. Or alternatively, not all organisms are pathogens, so it could be simply the ability of that organism to persist in association with your bodies and potentially even be helpful to your body. But that's all the ecology of the organism, and therefore it's all dependent upon the phenotype of the organism, although again, there will be an underlying basis that consists of the genotype of the organism. So that's some discussion of the difference between genotype and phenotype. As far as we're concerned in this course, our emphasis will be very much on phenotypic differences, but in microbial genetics there is a strong genotype bias, considerations of genotype. So the book then talks about things that are relevant to what's known as the central dogma of molecular genetics. Not microbial genetics, molecular genetics. And the key to saying molecular instead of microbial is not only you're talking about molecules by and large and the movement of information around a cell, as I like to say. But also we're talking about something that is more general than just bacterial genetics. By and large, microbial genetics is a study of bacterial genetics, although it's not 100% the study of bacterial genetics. But the central dogma of molecular genetics is applicable to both microorganisms and bigger things, to both bacteria and eukaryotes, and also archaea. And so what is this? This is DNA and its replication. This is the taking of information associated with DNA and converting it into information associated instead with RNA. That's a process known as transcription. And then you have translation, which occurs at ribosomes, which is taking the RNA found information, that's found in messenger RNAs, and converting that into polypeptides. Now, the process of converting DNA to new DNA replication, it involves phenotype, because there's certainly proteins and other factors that are involved in this process. But nevertheless, what you're doing in the process of replication is basically replicating genotype. The process of converting DNA-based information to RNA-based information, on the other hand, is the first step for most organisms, for organisms that have a DNA genome, of going from, in fact, let me back up, for all organisms, but there are some organisms that in the course of their replication go from DNA to RNA and RNA to DNA as a replication of genetic material. But the process, in most cases for most organisms, for all organisms, Going from DNA to RNA, the process of transcription, is the first step of going from genotype to phenotype. And in fact, in some cases, the RNA on its own provides phenotype, but mostly, at least in terms of how we understand and in fact teach this process, mostly it's the RNA that then gets translated, the RNA-based information that then gets translated into amino acid-based information, the polypeptides and ultimately proteins, that represents the key link to the production of phenotype. And then the proteins, in their catalytic actions as well as their structural actions and their ability to transport, etc., are the key underlying basis of phenotype. I like to go further than that. So you've got the molecular basis of phenotype, but all of that underlies the organismal level phenotype, what we see when we actually are looking at the organism rather than taking the organism apart and understanding its molecular details. So an example of an organismal phenotype in bacteria would be its doubling time, how long it takes to go from one cell to two cells. And it's the organismal level phenotype of the organism. Again, this is my perhaps somewhat unique way of looking at the world, but it's the organismal level of phenotype that actually interacts with the environment and thereby gives rise to the ecology of an organism. So you've got DNA. DNA can be replicated. DNA can be converted to RNA-based information in the process of transcription. RNA-based information that's found in what's known as messenger RNAs can be converted to protein-based information, first to polypeptide, then protein. In the process of translation, these proteins together in their actions give rise to an organism. I mean, there's more molecularly associated with an organism than just their proteins, but whatever is associated with the protein, with the organism rather, it is a product of some protein that the organism is associated with, that the organism produces. So you then have this translation gives you the proteins, and then the proteins give you ultimately the organism level phenotypes, and that the organism is what interacts with the environment. So replication kind of goes sideways here, but transcription to RNA, translation to proteins, production of phenotype that ultimately is the organismal level phenotype, which interacts with environments to give rise to an organism's ecology. So in the book, they go through some of the details of DNA replication, and that's not something that I'm going to talk about here. This is something that in the actual lecture that I give on this subject I talk about. But there's various kinds of proteins that are involved in DNA replication, and the process has some quirks to it that we like to talk about, but that's not what I'm going to talk about here. Instead, I'm going to move on to talk about specifically the control of gene expression. So in gene expression, what we mean is that we take some piece of DNA, which is the gene, and we convert the information associated with that piece of DNA into something that ultimately can have an impact on phenotype. And so this could be an RNA, but as noted, it's usually a protein. So when we express a gene, what we're doing is making a gene product, and in all cases that gene product is going to be a piece of RNA, but in many cases the gene product ultimately is going to be a polypeptide, which thereby is useful as a protein. So we can control the expression of a gene. We can control gene expression, therefore, either at the level of production of the RNA, which would be a transcriptional level of control, because transcription is how we go from DNA-based information to RNA-based information, or we can control gene expression at the level of translation, so that's going from the RNA to the polypeptide, Or indeed, we can control gene expression post-translationally. So whether or not the RNA is made in the first place, that's transcriptional level of control. Whether or not, given that you've got the RNA, you therefore make the polypeptide, and at what levels you make the polypeptide, that is a translational level control. And then whether or not the polypeptide is functional once you've made it in the cell, or how functional it is a post-translational level of control. So replication kind of goes sideways in this scheme. DNA begets RNA, begets polypeptides, begets some kind of function. Now we can go beyond that and say the function interacts with the environment and there are environmental feedbacks and blah, 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 but we're not going to worry about things going that far. So when we're controlling gene expression, yes, indeed, we can have a lot of mechanisms of control at the level of translation, and we can have a lot of biochemical mechanisms at the level of that are post-translational. But by and large, what we're going to concentrate on here is transcription level control. Transcription level control can be broken down into two basic types, what are known as negative control and positive control. So negative control means that you have some kind of factor that has the effect of decreasing the level of transcription. And with positive control, you have some kind of factor that has the effect of increasing the level of transcription. Now it's important to realize that increases or decreases in levels of transcription are going to be specific. It might be that you have a factor that controls the level of transcription of multiple different genes. However, we're not talking about, for example, doubling the amount of glucose in an organism's environment, which has the effect of increasing the rate at which the organism replicates, which has the effect of increasing the rate of all of the genes that are being expressed while the organism replicates. Or by alternatively, if you take the amount of glucose and cut it in half, that's going to reduce the rate at which the organism replicates and reduces the total rate of transcription of genes. Instead, what we're talking about are things that specifically, these are going to be proteins that bind to DNA in specific locations and thereby control the expression of specific genes. So how do they control the expression of these specific genes? So again, negative, positive. If you have a negative control, it means you binding to the DNA and it's preventing or decreasing the rate of transcription. If you have positive control, it means it's binding to the DNA and you're actually increasing the rate of transcription. So how does this happen? The answer is something called RNA polymerase. So RNA polymerase is the protein that is responsible for transcription. It polymerizes RNA. Technically, RNA polymerase is a DNA templated RNA polymerase. So the DNA, the sequence of nucleotides that make up that DNA, represents the sequence of a gene, and the job of the DNA-dependent or DNA-templated RNA polymerase is to read what's known as a reading frame of a gene. And that the process that results from that RNA polymerase is you take a linear sequence of nucleotides of DNA and you convert it into a complementary, as they say, linear sequence of nucleotides of RNA. Complementary because every adenine is going to give rise to a thymine rather than another adenine. Every guanine is going to give rise to a cytosine, every cytosine a guanine, every thymine an adenine. Although in going from the DNA-based information to the RNA-based information, the bases that are found are actually changed where the thymine instead is a uracil. So A gives rise to U rather than giving rise to T. So it's the RNA polymerase that is responsible for transcription, And therefore, it's control of the action of the RNA polymerase that results in control of gene expression at the level of transcription. So if you're able to block the RNA polymerase, then you're not going to get transcription of a gene, and therefore no expression of that gene. On the other hand, if you're able to increase the likelihood that an RNA polymerase is going to transcribe a gene, then you'll get more expression of that gene. Well, that's still control of gene expression, but in this case it's control that gives rise to an increase or a positive result. So that's positive level control. Whereas if you're blocking the RNA polymerase, that gives rise to a negative result. So negative control is blocking the RNA polymerase, and positive control is encouraging, essentially, the RNA polymerase to transcribe a gene. And it's important to start out by talking about the control of gene expression in those terms, because when you actually look at the control of gene expression, it can get very confusing very fast what actually is positive control and what is negative control. So without going into the details, remember that if you have the factor and it binds to the DNA and it prevents the RNA polymerase from transcribing a gene, that's negative control. It's a negative effect on gene expression. On the other hand, if you have something that binds to the gene and actually encourages the RNA polymerase to transcribe a gene, that's having a positive effect on gene expression, and therefore that is positive control. Okay? So, when we get to the nitty-gritty of what's going on, it gets more difficult, and that's because there are other factors that are involved in controlling whether or not the primary factor is able to bind to the DNA. So I guess we could draw this out. So we have DNA, and ultimately this gives rise to RNA. In order for that to happen, you need RNA polymerase to do its thing. So in the middle here is RNA polymerase. Now, if we have negative control over RNA polymerase, then you have some factor, we'll call it R for regulation, which has the effect of blocking the RNA polymerase from making the RNA. On the other hand, if you have positive control, in fact, I'm going to come back over here, and instead of an R, I'm going to put an N, negative control. That's a factor. That's a protein that is able to bind to DNA, clamps onto DNA. Alternatively, if you have positive control, which we'll use a P now, that's going to have a positive impact on the ability of the RNA polymerase to transcribe the gene. And hence I put a check instead of an X. So how does the positive control work? The positive control works by encouraging the RNA polymerase to bind. The RNA polymerase is not going to just bind to any piece of DNA and start transcribing. It needs to be encouraged to bind to the DNA. And one of the things that encourages the RNA to bind is the positive control factor. So if they bind to the DNA, then that encourages the RNA polymerase to bind to the DNA. And if the RNA polymerase binds to the DNA, then you get an RNA product. Alternatively, if you have the negative control factor bind to the DNA, it's going to interfere with the action of the RNA polymerase, and you're not going to get a product. So you can think of the negative control element or factor as being a dam. And I'm trying to think of a nice analogy for the positive control. It's difficult for me to think of it. It's basically something that's attracting the RNA polymerase to bind to the DNA. But definitely the negative control is a dam. Okay, so now we've got the problem where it becomes more complicated. It's relatively easy to understand this from the perspective of positive control. You'll have a factor and there'll be something that stimulates the factor. It might be the production of new factor in an active form and it simply binds, or it might be that the factor is produced in an inactive form and has to bind to something else in order to be activated in order to bind. So we'll call that PA for active, and we'll put it up here. So it has to be in an active form in order to in order to bind to the DNA and thereby encourage the RNA polymerase to bind. It might be that this factor is initially produced in an active form, but it might also be that the factor is produced in an inactive form. I'm putting that as PI. In which case, it has to interact with something else. We'll call it L for ligand. And that's what converts it to an active form. And in an active form, therefore, it's able to encourage the binding of the RNA polymerase. But alternatively, you certainly could have a circumstance where your initial production of I'm doing this backwards, where your initial production is in an active form, but that might, as a result of binding to some different ligand, convert it to an inactive form. In this case, when the factor binds to the DNA, after binding to the other factor, the ligand, the small molecule that's affecting its function, it would fall off of the DNA. So if you had control that was such that the factor was active only when you had another substance around activating it, then you would only get DNA binding when you had that other substance around, in which case you would get gene expression when the other substance was around. Alternatively, if the other substance had the effect of inactivating, hopefully I'm saying this correctly, consistently, then when the substance is around, it would cause the activator, the positive control element, to fall off of the DNA, in which case the RNA polymerase would no longer be encouraged to bind to the DNA. Let me attempt to state that in a way that I'm sure I'm saying it correctly. So we either could have something starting out active, and then it simply, there's no further control, in which case you make the thing